Kamaraj University, Madurai, since 2005. She handles classes with passion in the areas of immunology, lab in immunology, molecular diagnostics, animal virology, human health and diseases. Their team is working in the area of human immunogenetics, trying to understand the inter-individual differential disease susceptibility by exploring the natural killer cells of the individuals. She is the recipient of Innovative Teacher Award presented by Madurai Kamaraj University and a gold medal winner in the year 2009. She is also a recipient of Raman Postdoctoral Fellowship for the year 2016 to 2017 by UGC under Indo-US Knowledge Exchange Initiative at the University of California, San Francisco, USA. She is also recognized as a mentor by DBT, Government of India, for motivating students for pursuing basic sciences. She did her PhD program during the year 2000 to 2005 by availing JRF and SRF from CSIR, New Delhi. Dr. Jayalakshmi ma'am has completed her BSc and MSc Zoology from Holy Cross College, Nagarkoil, by securing university first rank and gold medal from Manonmaniam Sundaranar University in the year 1997 and 1999. Thank you for joining us, ma'am.
Yeah.
Over to you, ma'am, please.
Okay, thank you. Thank you very much, ma'am. Thank you for the introduction. And good afternoon to one and all. And after a lunch, you are all here, joined for this webinar. And so it will be a lighter session about the topic that is of much interest, which is on hot debate in every media. Okay, so hope you will enjoy it. We will be discussing about the past, present, and future trends in vaccinology. So greetings from the Temple of Learning, Madurai Kamaraj University. And my lecture outline will be, I will be sharing some background information, why or what is the need for this vaccines, and then about the birth of vaccination. So that is why I'm concentrating about the past and development of vaccines. Then various types of vaccines, vaccine efficacy, as well as the herd immunity. And India leads the vaccine market. That's the interesting information for every one of us. Then about so that will be the present aspect of vaccines and the myths and facts of vaccine. Everybody should understand it and we should disseminate to people. And so we should be a propagator for vaccination. We should be for vaccination, but not against vaccination. But there too, we should know our limits, right? And so you, all of you will be much eager to listen about the SARS-CoV-2 vaccine and what will be the future of our vaccines. So how we are interested to have our vaccines, okay, further. Okay, so actually we are imagining, we are living in a world expecting that a perfect world, that is where there is a clear discrimination of the pathogens from us human beings. So if we are living in this way, can you imagine like anyone who is living in their own micro environment, they will not be worried about others. But when you are all mingled together and competing for resources, then you will be having some of the some tough time together. So that is what actually in the real world also things are happening. We are along with the pathogen. So pathogens are also one among amidst us, and they want to spread their diseases, like they want to multiply. That is why they are causing the diseases. It is not as such they want to cause the disease. It is like they want to spread the disease because they need to multiply, and that multiplication they need some host or environment, and that is unfortunately for some of the pathogens it's us. That is the reason like we are facing this difficulty. So pathogens are almost present everywhere. We cannot say this particular area is free from pathogen and this is not. At least to a certain extent you can control your environment, and by having some of the some of the devices, sanitizers, sterilizing equipments, all those things. Certain area only you can do, but you cannot entirely do to the globe, right? We cannot do. So this picture shows the dispersal of Mycobacterium tuberculosis. If a person with Mycobacterium tuberculosis inside, if they cough or sneezes, what happens during their cough, this is it okay? Is there any problem?
Ma'am, no, ma'am, you're audible now.
Okay, okay.
Yes,
okay. And the pathogens, there is a dispersal of Mycobacterium tuberculosis in the entire atmosphere, and everyone who walks through that area, we are coming across it. So this is the situation in India. So if we are traveling in a public transport, it is for sure we are going to come in contact with some or the other Mycobacterium tuberculosis. That is why on the day of birth itself, the Mycobacterium tuberculosis BCG vaccine is actually given. So rather than coming in contact with a very much vigorous pathogen, we are going to give an attenuated form of the pathogen, and that is actually going to promote the immune response. So different countries or globally, the World Health Organizations, they are trying to develop initiatives to reduce the disease burden. So in this, vaccination comes the first. So vaccination is really a boon for the society, which has been given by the medical society, to prevent the infectious diseases. Then comes the mass treatment, then followed by sanitation, vector eradication, intermediate host eradication. So what is the vector eradication? For example, mosquito eradication. What is intermediate host eradication? Sometimes the pathogens, they are harboring in some other host, some of the snails. Okay, some of the snails, they are sources for some pathogens. Likewise, they can go. Then we should have very good diagnostics. Okay. For anything, the prevention is better than cure. That is a proverb what we have learned from our school days itself. So the vaccination comes the first. Okay, so in this lecture we are going to discuss about the vaccination. This picture and slide will describe you the principle of vaccination. So what happens when a disease causing organism infects a person? The person can respond to the pathogen in a different way. Likewise, think there is a person telling a harsh word to you, or a person A to person B is telling, and the person B can take that word in different mentalities, right? In a he or she can ignore it, or she can react to it, or he or she can cry over it. So there are different ways. Likewise, we are also, when we are exposed to the pathogens, disease causing agents, we are responding different way. That is, one way is responding in a better way and reaching in recovery. The another way is responding and there is poor response and it leads to death. And sometimes, okay, there is response, but it can lead to disability. For example, the case of poliomyelitis. And what happens in the recovery? We are not interested now with what happened in the death individual or disabled individuals. We are interested in recovered individual. Okay, so everybody likes the smiley of really a smiling face, okay, rather than an angry face or a sad face, right? So we have it in our keychains and we enjoy carrying it always. And what is really happening in this recovery phase? When this was actually studied, the immune response was studied in those individuals. They could identify immune memory, a memory concept. So that memory concept is being used. Okay, that immune memory concept is being used in the development of vaccines as well. Okay, one is that immune memory. The another is, you cannot give this may be a luckily one in 100 individual who can respond to a live or viral strong pathogen. but so it cannot happen. We don't want to lose people. Okay, so what is being done is we are attenuating the antigen. So attenuating the antigen, in the sense here it is the pathogen. Okay, we are attenuating the antigen, and that is actually known as the vaccination. So here we are attenuating the virus or pathogen, and we are injecting into an animal. When it has been injected, there is no disease development, and that leads to immunity, and this person will develop immune memory now. So you have given a weak dose of antigen. Okay, weak dose of antigen. So this analogy you can compare. You are staying in a bus stop one particular day. You are seeing the thief coming and trying to steal away the pouch from some of the other person. He hasn't approached you. You from a far side, you have seen he is approaching some other person and he is running away. The person's picture comes very strong to your mind, and for the next day, after 1 week or 1 month or 1 year, if you are seeing the same person approaching you, you will be very cautious, right? You will be very cautious. You will be holding your pouch or something. So that is how you are recovering it. That is by your memory, your photographic memory of your brain that brought back the picture of the thief. Okay, so here the immune memory is recalled back when the same antigen, not the attenuated form of antigen. Attenuated form of antigen was given for the first time, that is the vaccination. For the second time, direct antigen is approaching you, and immune memory is there, and it is actually protecting us. Okay, so we are all living in a different environment. Everybody is being subjected to various pathogens, but not all the infected will develop the disease. Everybody can be getting infected if we are coming in contact with the pathogen, but not all the infected develop the disease. This is a very important immunological dictum. Okay, so this will be and how it is. This is because of your various aspects you can recall for this not developing a disease. It is, you can attribute it to your positive mindset, it to your healthy body. Then you can attribute to your good immune response genes, what you have inherited from your parents, all those things. Okay, so further the principle of vaccination relies on memory cells, and then followed by, whenever next time when the antigen comes, you have an amplitude of antibody responses there, and that response will be really good and useful. So what are the early attempts at immunization that was done? Humans from the beginning itself, initially they were hunter-gatherers and they were trying to fight their lives. Okay, they were running from tree to tree, they were jumping, they were hiding in some places, afraid of so many wild animals. Then they started to build some tents, all those things they tried doing it. Then after they tried to settle in certain places and then started to for irrigation and agriculture. At that time, now they wanted to develop their community as well. Okay, and when they were living in groups, the diseases, infectious diseases started to emerge, and these diseases they wanted to control. So long back, before, you might be knowing that Edward Jenner has contributed in around 1776 or sometime. He has contributed to prepare the vaccination against or demonstrate vaccination against the smallpox using a less pathogenic form of cowpox. Using cowpox, he has developed it. But those days, the early days, they didn't know much about the pathogen, right? Pathogen as such, what is causing the disease. They knew about the diseases. So before even Jenner, like in India, China, all those regions, what people tried was they took the dried matter from the pustules of smallpox, and they will be putting it, inserting it into the nostrils. Okay, as you can see it in the picture, like they tried inserting it into the nostrils. And what happens in the birth of vaccination? So this is actually, vaccines are widely recognized as one of the greatest medical advances in human history. And this is a very interesting experiment which has been demonstrated by Edward Jenner. On 14th May 1796, Edward Jenner used cowpox infected material obtained from hand of Sarah Nimes. Who is Sarah Nimes? Sarah Nimes is a milkmaid from his home village in Berkeley, and it is at United Kingdoms. Okay, he successfully vaccinated an 8-year-old boy, that is James Phipps, and like he tried inoculating him. Afterwards, then further after inoculating the lesions from the cowpox from the Sarah Nimes' hand, he tried really with challenged multiple times with smallpox pustules. This boy didn't develop disease. Okay, he didn't got infected and he was not suffering from the disease. So he tried publishing it or putting it on a forum, but people did not accept it immediately, this concept. But at that time, Jenner didn't know anything about the virus or about the pathogen as well. They knew at those times about the diseases, not the pathogens. Okay, then further when he was challenged, he didn't lose his hope. He tried with his own son. Okay, that concept now you have it in the Russian vaccine also against SARS-CoV-2. Putin, he has told he has his daughter is being vaccinated. Okay, daughter is being vaccinated against the new SARS-CoV-2 vaccine. Okay, so as an emotional strengthening to give the public, this person has done it to his son itself, and then to other people, and further, you know, this it has been completely approved, and now the smallpox is completely eradicated from the world in 1980s, and this is the statement from World Health Organization. Okay, so the world and its people are free from the endemic smallpoxes, and the last reported case of smallpox was in Somalia in 1977. After that, there is no case. So it is, you can see the next, this is a map, world map, to show you the different countries at which time they have actually eradicated or completely have seen the last case of smallpox. In North America, it is 1952. In South America, it is 1971. In Africa, it is 1977. And in Asia, it was 1975. And that was in Bangladesh. Okay. And in Europe, it was in 1953. So you can see here, the developed countries, this control happened at a quicker pace than comparing the developing countries. Why this reason? Maybe because of the density of the population, and then because of the affordability of the world. Okay, so now coming to the vaccine development, this, as we know, vaccines have proved to be a boon for the prevention of infectious diseases. So and it is actually a cost-effective measure for preventing disease. It is actually diminishing the morbidity and mortality. It is reducing economic burden, and all this should begin with some basic research. Okay, so this basic research, everybody will be contributing, and we are also contributing to the basic research by studying the various infectious diseases and understanding the immune response, what is happening between the differentially susceptible individuals. Okay, and what will be the ideal vaccine for anything? You will tell ideal teacher, how the ideal teacher should be. You have some characteristics put forward. An ideal student, you have some characteristics put forward. Likewise, you have ideal vaccine. So it should stimulate fast protection within 2 weeks. There should be needleless administration. The administration should be in combination. It should elicit high level of protection, and the protection should be to all age groups, and it should be free from storage. But to our understanding, none of the licensed vaccine have all these characteristics together. They may be having few of the characteristics together. Okay, so it is not a must that a vaccine should have all these characteristics, but at least some of the basic things it should have. Okay, coming to the types of vaccines, we are having various types like live attenuated vaccines, and here you are taking the disease causing agent and culturing it in some suboptimal conditions, and that will make it attenuated. Okay, and afterwards it will be purified, some additives will be added, and this will be used as vaccine. Examples are measles, mumps, rubella, varicella zoster. All these are done this way. Next comes the inactivated vaccine. Here you are using the disease causing agent is being treated with some chemical or thermal treatment, and by this way it becomes inactivated. And this inactivated, you can see there is a into mark here, so it is actually killed, and now it will be purified and the additives will be added and it will be used as a vaccine. The example is hepatitis A, influenza, pneumococcal polysaccharide, all these. And then the recombinant subunit vaccine, that is here recombinant subunit, that is it is used or created using recombinant technology. And subunit, one small part of the virus or pathogen will be taken and it will be fused with some other expressing system, and then it will be allowed to express the proteins, and this will be purified, added with the additives, and this will be used. And a classical and beautiful and successful example is the hepatitis B vaccine. Okay, here the surface antigen is being recombinantly expressed and it has been used as a vaccine. Then comes the toxoid vaccine. So everybody is much familiar about this toxoid vaccine. What commonly you call is the TT vaccine, the tetanus against, that is actually the tetanus toxoid. Here we are not causing the disease causing agent. The disease causing agent produces the toxin. That toxin will be taken and that will be subjected to chemical treatment, and now this will become inactivated toxin. This will be, to this it will be added some additives and it will be used as a vaccine. Example is the tetanus as well as the diphtheria vaccine. Next comes you have some of the polysaccharide vaccines. Here you have some difference between the child versus adult immune memory. Okay, when a protein vaccine is being, so in this you should know which will be a best source of vaccine is generally the proteins will be the best source of vaccine, or and the chemical complexity based proteins will be best. So they can actually induce humoral response and immune memory in both in infants, and this will also induce humoral response and immune memory in adults. So a protein vaccine, if you are giving in both in infants as well as in adults, it is stimulating the immune response. But when polysaccharide vaccines are actually given sugar based, when it is being given, it is for right away it is helping in human adults to develop the immune memory, but in the children there is no immune memory is being developed. So how can you come across it, overcome it? What will you do? In our basic theory classes we have read that if you have to make a polysaccharide to become immunogenic. Okay, immunogenic, that means capable of stimulating the immune system. So if you want to make it, what you will do, you will compare it, you will combine it with the carrier molecule, and those carrier molecules are generally larger molecular substances like the bovine serum albumin or O-albumin, like that. You will combine it with other protein carriers and then you will give. When you give like that, it will be producing. Okay, when it is being combined with carrier, then it will be producing. And using these approaches, so various vaccine preventable like diseases has been worked upon, and more than 135 successful vaccines are being produced. Okay, starting from 1798 for the smallpox, then it goes on, then you can even add to 2020 for SARS-CoV-2 vaccine. So this slide goes for some 4 slides together, it runs. So more than 100 against 135 diseases, the vaccines has been produced. Okay, so the cumulative number of vaccines developed since the first vaccine in 1798 by type are mostly at the earlier time. You can see the killed vaccine, like they were being produced or attenuated form of the alternate from alternate host, they were being produced. Afterwards, now when you come to shift over to the different centuries, you will see different types of vaccines are being added, and by protein conjugate vaccines, polysaccharide vaccine, purified protein, toxoids, recombinant protein, plasma derived, like that many different types, using multiple approaches people are producing. Now even for SARS-CoV-2, if you check, there are some vaccines which are produced based on mRNA, some vaccines based on its surface antigen, some based on the inactivated virus, like that different forms are being produced. So why actually, why does my choice matters to others? Okay, so if you are interested to take a vaccine, if you are interested, yeah, you can take. But if you are not interested to take, will it affect only you or even others? It is going to affect, yes, it is going to affect others as well. That is this concept can be explained by using the terminology herd immunity. Okay, so this term also, now every layman is familiar with this herd immunity. Thanks to the media, they focused, everybody will be questioning the doctors of when will we reach the herd immunity status, when will we reach the herd immunity status, like that. Okay. So here, when no one is immunized and actually the disease is, if the disease is spreading through the population, what happens is the disease will spread much at a faster pace. Can you see the red number increases very fast? But when some of the population is immunized, there will be still disease spread, but at a little slower pace when compared to that of the previous. But when most of the population is immunized, and then the disease spread will be at a slower, very slow phase. And this immunization, we can achieve it by 2 ways. One is a natural way of getting immunized. How? By acquiring the disease and we develop antibodies and we are immunized. And second is by the way of vaccine. So when we are expecting this to happen by its own, if it is a strong and effective pathogen, we are going to lose so many lives. That is why we are aiming for vaccines, and until the vaccine comes, we are trying to maintain some social distance, and we are trying to have some mask and using some soaps and soaps to wash our hands and all those things, right? So the disease transmission in a non-immune population is quick, but it is if there is a herd immunity, that is, if one or two or few of the individuals are immunized, then the spread will be controlled. So this immunity passport, what is immunity passport? So the based on whether the person is being whether a person has developed the SARS-CoV-2 virus infection, based on that, they originally thought whether we can give passport stamped as SARS-CoV-2 immunity passport. But no, that is not a good idea, because the chances of reinfection is also there, as well as this virus will be very fast mutating, and it will be producing new strains, and this cannot offer a lifelong protection. So this giving an immunity passport is not a good idea. There are so many diseases in this world, and for everything we don't need vaccine, right? So why do we need for a few of that? Because of its RO of infectivity. So what does the mean by RO of infectivity? or R naught, value of infectivity, the number of people that one sick person infects on an average is called R naught. So the maximum R naught values for a few viruses are, it's very high for measles, that is 18, more contagious, followed by mumps. Then followed by that, comes the SARS, and then HIV, Ebola, hepatitis C, all those things. So for SARS, the number is around 3 to 4. Okay, 3 to 4 persons can be infected. One sick person can infect on an average. Okay, so based on that, the R naught value is calculated, and for this we need the vaccination to arrest this. Generally, to develop the natural herd immunity, in England they used to celebrate the chicken pox party as like a birthday party. If children, they are if any household children is being infected with chicken pox, then the neighboring area children will be invited to the house and they will be celebrating as a party, because if chicken pox is affecting an individual at a young age, it is not going to cause much deliberative effects, rather than if it is causing at an older age, it is going to cause more serious infections. And all this what we are aiming is breaking the chain of transmission, whether it may be the natural herd immunity or artificial herd immunity. The herd immunity thresholds for some diseases. So this naturally, depending upon the contagiousness, the threshold of the herd immunity will be varying. You can see this way, it is varying from 75, anywhere between 75 to 94%. What does that mean? If 94% of individuals are being immunized, then only, that may be natural or artificial. If they are immunized, then only the herd immunity threshold will be reached. Okay. So, but for this SARS-CoV-2, we haven't yet arrived with a threshold calculation. Okay. So the level of vaccination coverage is required to interrupt disease transmission will actually depend on the ease with which a disease is transmitted and the effectiveness of the vaccine at stimulating the immunity. Okay, we have produced a vaccine. Now we want to test the vaccine efficacy. How can we test? If we know the attack rate of the disease in an unvaccinated group as well as in a vaccinated group, then we can calculate it. Okay. So the attack rate in unvaccinated minus attack rate in the vaccinated, that you will put on the top, and the bottom you will put attack rate in the unvaccinated, and you will be making it into percentage. Then you are getting the efficacy, vaccine efficacy. And the observed vaccine efficacies of some of the vaccines are, it is ranging from 70 to almost 100%. Almost 100%. Okay. For pneumococcal polysaccharides, if in the case of adults, it is only 70%. But for meningococcal, conjugate meningococcal or human papilloma virus, HPV, then hepatitis A, for all those, it is 100%. Okay. What is the efficacy? This efficacy is actually going to do good, and if the efficacy is low, people will start to try to improve the vaccines. Okay. Always, sometimes the common reactions happens to vaccines which are routinely used in several industrial countries. What will be the reactions that will be happening after vaccinating our children? We might have experienced they will be crying with pain, there will be swelling, there will be redness, there will be a mild temperature, fever, and sometimes some systemic symptoms on the body. Okay. So these adverse reactions, you can call it as these adverse reactions. Some reactions are very common. Okay, for every child, there is a mild rise in temperature, there is a pain, and the child has fever. sleepless night for one particular day, and then next day it becomes all right. And so there are some symptoms persistent infection or persistent fever, or sometimes some rashes. Those are all sometimes unusual symptoms. And sometimes you might have heard, after taking a particular vaccine, this child is suffering from some of the diseases. But those things should be proved properly. Okay, that we will be discussing in the myths and facts. Okay, and the safety versus efficacy of the vaccine type, if we are comparing, there is a replicating virus group as well as non-replicating virus group. What is the replicating virus group? If you are using a live attenuated vaccine, or you are using a Jennerian vaccine, that means from some other animal infecting or milder form of the disease pathogens you are taking, or replicating vectors if we are taking. So these are all replicating vaccine types, and non-replicating will be completely inactivated virus, or virus like particles you are giving, or some subunits you are giving, or some nucleic acid based you are giving. So the safety wise is, which is very excellent, the nucleic acid based. Okay, why? Because they will not revert back. But the efficacy wise, which is very good, the replicating type of vaccines, they are actually very good in the efficacy, or say the efficacy wise. Okay, so the success of this live attenuated vaccine is really effective, because they can actually replicate, deliver sustained dose of antigen, and replicate at the exact anatomical site of infection, whether it may be a throat or whether it may be a esophagus. Like that, it reaches the site of anatomical site of infection, and there will be the immune response happening. But it is not, however, it is not advised for immunodeficiency patients, because occasionally they may revert and they may cause little severe diseases sometimes. Okay, so that we will see in the case of polio what is actually happening. Okay, but anyhow, the due to the thanks to the vaccination, the incidence of viral diseases has come down, whether it may be a poliomyelitis, measles, rubella, or mumps. After the year of introduction, it has come down very well. Okay, and in India, in India, in the last case of poliomyelitis was reported in the 2011, and from that time no case has been reported, and this case was reported in West Bengal. Okay, West Bengal. And the last the environmental sample was identified in 2010. And what is in the environmental sample? That is, they will be checking in the drainages. Okay, drainages. That is, this virus poliomyelitis, this spreads through fecal-oral route. So if a infected child is using a toilet, the laboratory will be having the virus. So, and if the hand is not washed proper, and if they are playing with some fomites, then they can be a chance for contamination, and a chain of spreading can happen. So however, our neighboring countries are not completely free from polio. So we should be very careful at the borders, and we should always continue to give the oral polio vaccine, even though it has been completely eradicated from India. The different state-wide immunization programs have been started, and every state is following it. And the Kerala was the very first successful one in 1993, followed by Tamil Nadu in 1994. Okay, and sometimes there was a back. You might have read about the news. There is a pertussis, incidence of pertussis occurring back. So we will know that the vaccine schedule will be generally given before the child steps into the school, right? So the mother will be very cautious. Until that time, the child is always a child, pampering so much, taking care, everything. But after the child goes to school, the parent also tend to concentrate on their studies or other things. They tend to forget. The DPT booster doses are generally given at when they are in the first standard, when they are going to the first standard, around 6 years. At that time, there are some child got missed this vaccine, and the diphtheria percentage shooted up, and there was also deaths. And when that happened, the government said, okay, repeat giving booster doses, and it was given back in the schools. Okay, the central government has launched a scheme called as Mission Indradhanush, and Indradhanush stands for rainbow, and in rainbow there are 7 colors, and this program is protecting children against measles, polio, pertussis, tetanus, TB, diphtheria, and hepatitis B. Okay, so if any child has been missed with these vaccines, they can be taken care, and it has been they are being vaccinated. Okay, what will be the different vaccine production methods? You can produce vaccine, manufacture it by in vivo, in vitro, or chemical synthesis. Okay, sometimes any one method you will follow, or all the 3 methods you are going to follow, like that. Likewise, using this, there will be different generations of the vaccines. As we are having our latest generation mobile phones, originally it was of button kind of phone. Now you have touch screens, and so the functions are made easily. Likewise, in vaccines also, the different generations of vaccine are there. Let me take the example of hepatitis B vaccine for explanation purpose. And hepatitis B vaccine, this is actually originally in 1981, it was a plasma derived vaccine. What is meant by plasma derived vaccine? Now people are trying to do the plasma therapy. They are for SARS-CoV-2, they are trying to isolate the antibodies from the recovered persons, and they want to give it to to whom they want to give, the critically ill patients, whom who are not able to synthesize their own antibodies. Likewise, here the antigens, they were harvested from the infected persons, and that was used as in vaccine. And this was discontinued in 1990, and then you have second generation vaccine. These are the recombinant HBV vaccines. This does not contain any blood products from any other individual. So you won't develop the disease from a infected individual. What is being done? The hepatitis B virus is purified, genomic DNA is extracted, this DNA is cloned into a bacterium, and the S gene from the bacterium is isolated. S gene is the surface antigen, the outer core, outer surface layer antigen is being isolated, and it is being used for the construction of the plasmid. And now you have the insertion of plasmid in the yeast is done, and the master seed is being made, and this is being amplified in the yeast cells. So this is free from human contamination. By all these things, the great good news is the India is leading the vaccine market, and more than 2/3 of the total volume of vaccines manufactured is exported, while the rest is utilized domestically. In 2007, about 33% of the vaccine market in the world was contributed by India. And India is the largest producer of recombinant hepatitis B vaccine as well as the Nasovac. And now in 2019, GlaxoSmithKline, they represented the largest player in the Indian vaccine market. And by 2025, in 2019, the vaccine market worth of India was about, in INR, it was 94 billion, but it is expected to reach 215 billion in 2025. Okay. So what are the different stages you should cross for producing the vaccines? There are different stages like phase 1, phase 2, phase 3, and then you get the license. So what is phase 1? Here it has been, the vaccine prepared has been tested in few individuals. Even before the phase 1, the animal studies will be done, right? And after this phase 1, only in few individuals. In phase 2, if it is okay, then they will pass on to phase 2. It will be done in 50 to 100 individuals. And then phase 3 in 1,000 to 10,000 or more individuals. And then only the license will be applied or given. Okay. So these are the 4 stages. So every stage it should come across, cross, and then only it will be given. So there was a promise given in August 2015, the Indian vaccine will be launched ready for public use. But there was a huge cries from the various people. How is it possible to do it in a very short time period without crossing all these phases of trials? But after the announcement only, the registration of phase 1 trial itself started. Then how is it possible like that? So now our vaccine is being tested, but it is not yet ready. Okay. After even the vaccine is being produced, every batch of vaccine is tested for quality as well as the safety. Once the vaccine is approved, it continues to be tested. Even after it has been approved, it will be continued to be tested for its potency, for its purity, for its sterility. And then FDA review the results of these tests and inspects the factories where the vaccines is made. And this will help sure the vaccines meet the standards for both quality as well as the safety wise. There is some simple technique used to monitor the vaccine, that is called as vaccine vial monitor. This is a sticker kind of thing where you paste, where you will be pasting, and depending upon the color change, if the vial is being subjected to increased temperature or unfavorable temperature, then the color change will be happening, and based on that you can tell the vaccine can be used or not. Okay, so still there are so much of promising areas to explore, like cancer, leprosy, HIV, Zika, malaria, Ebola, and adding to that you have also SARS-CoV-2. There are there is large scope for this vaccine research industry. And what will be the risk of vaccines? Inactivated polio vaccine replaced the live oral Sabin vaccine, and because of its risk, sometimes it is you should know that in the developing countries only the oral polio vaccines is given. In developed countries, it is actually by injections only. Completely inactivated polio vaccines are only used. And then the acellular pertussis is actually causing the high fever. Then there is a saying that MMR, that is measles, mumps, rubella vaccine, it is actually precipitates autism or inflammatory bowel disease. But neither we have biological or epidemiological evidence to defend this point. Okay, this causes autism. We don't have proper evidence. In developing countries, adverse reactions are acceptable price for prevention of death and debilitating diseases for the masses. So as we are having when the population is very high, then vaccine will be the best and good choice. Okay, what will be the social issues for the new century? Safety concerns and rise of vaccination. Now the trend is they are developing hashtag against everything. So if a vaccine comes also, people will start, okay, hashtag for no vaccine, hashtag for no vaccine for the indigenous vaccine of India, or from which is going to come from abroad. Like that, people can have different voices. Okay, their thought may be different. And but for the cost of developing countries, it is really high, as well as the adequacy of supply is also a question mark. Okay, for the 2 slides, I will be discussing about the myths and facts of vaccination. So vaccine cause autism, but we don't have any proper relationship like that, then it's not necessary to vaccinate so early in life. Why should you do it for the first day itself? No, we should. Vaccine is preventing the disease in most of the deadly diseases in very young individuals. So early in life itself, vaccine should be given. The vaccine schedule is too aggressive and should be spaced out. It's very close. We should space. No, it is like that. The decades of medical evidences are showing that the window period of the time, optimal time of window is given, and it is okay to follow the schedule. And vaccinations cause the diseases that they are meant to prevent. Vaccines don't cause the diseases, and here's why: because they don't contain active viruses. Vaccines contain unsafe toxins. No, it's true that vaccines do contain trace amounts of formaldehyde or mercury or aluminum, but those levels are very low when compared to our daily life habit, what we come across with these of the chemicals in our day-to-day life. We know that formaldehyde is really not good for health, but we are not sure whether the fish or preserved meat what we are taking is free from formaldehyde. Okay, and the effectiveness of vaccination has never been proven. It is scientifically proven things will be only come for vaccinating. So then there is another myth that is not vaccinating my child affects only my child. So some of the religions, they say God is there to protect us, and so no need for vaccination, and our child will not be vaccinated. No, it is not like that. Unfortunately, the one child, the breaking the chain will not be there, and fast the disease can spread. Okay, so with that we have finished the past and the present. Now we are going to discuss about the future of vaccines. That will be the future of vaccine will be the vaccines that have strong immunogenicity without increasing the reactogenicity. For that, there is lot of scope and long-lived efficacy. Example, measles, lifelong immunity is there. Like that, for others also, we are expecting long efficacy. Then cost effectiveness, that is storage at room temperature. For that, we are expecting. And mode of vaccine administration, that is non-invasiveness. People are looking into for what is non-invasiveness. That is the point. That there comes the edible vaccines. People are trying to produce vaccines with the help of the banana, tomato, potato, corn, rice, spinach, all those things. What they are doing in a simple way, the leaf or the plant part will be cut, and that will be fused with the pathogenic the important gene of interest for what we want to vaccinate. And now this fused will be set up. This will be allowed to grow, and as a callus it will be allowed to grow, and it is being growing. Now this, when it has been producing, they can produce or secrete the vaccines. But these vaccines, none of that has come as a very successful one. It is all tried. So many vaccines are there trying. Why? Because we have vaccines for some of the diseases, but it is not affordable by everyone, and people need non-invasive methods. And so this kind of research is going on. Okay, finally, to sum up, we have to discuss about the SARS-CoV-2 vaccine. So the SARS-CoV-2, as we know, there are various types of vaccines. So different forms of the virus you can take for developing. One can be a live attenuated vaccine, viral vector-based vaccine, a recombinant protein-based vaccine, that is the spike protein, and then DNA-based vaccines, mRNA-based vaccines. All these types of vaccines are tested by different countries throughout the globe. And one example here, what has been explained is the SARS-CoV-2 spike antigen. When it is being taken, when it has been injected, immunized to an individual, this antigen will be presented by an antigen presenting cell, and it will be presenting to a naive T cell, and this naive T cell will now transform that is to activated CD8 or CD4 T cell, and this can help in the elimination of infected cells, or this CD4 T cell can help the B cell, and this can help towards the formation of memory as well as the plasma cells, which will be secreting the antibodies, and this way also it can help in the clearance of the pathogen. Okay, so as of the March 2020, the WHO, they have given large list of the different laboratories who are actually working on various aspects of the viruses, as we saw in the earlier slide. Among that, the most successful or promising candidates the entire world actually was looking forward was AstraZeneca from Oxford University, Moderna from National Institute of Allergy and Infectious Diseases, USA, and Pfizer from Germany, and CanSino Biological from China. So these are in the different trials of 1 and 2 trials. Okay, but from Russia, first they told, yeah, our vaccine is ready. So what they have used is adenovirus that is actually used as a vector, and they have made adenovirus to express the surface spike antigen of SARS-CoV-2. They have engineered it such way, and here 2 doses are required, primary dose and 21 days after a boosted dose. And now you know what is happening in the entire scientific community is towards them, what they are going to tell, and whether it is effective, what is the efficacy, all those things. But really the efficacy and all those things are not yet tested. Okay, so with this, the references what was used for this lecture was the World Health Organization Vaccine Facts Book, the edible vaccine. There is a review from Austin Journal of Nutrition and Food Sciences, and the WHO draft for COVID-19 vaccines. This tells about the SARS-CoV-2 vaccines, and the mRNA-based vaccine, which was released from New England Journal of Medicine. Yeah, with this, the recap is we have discussed about the background and the birth of vaccination, development of vaccines, about the type of vaccines, the vaccine efficacy as well as the herd immunity, the R0 value. Then India is leading the vaccine market, myths and facts of vaccine and SARS-CoV-2 vaccine, the future vaccines, and about the edible vaccines. All this we have discussed in this lecture. Okay, so with this, vaccination has come a long way since Jenner, and we can be confident that vaccinologists will continue to extract beautiful melodies from the orchestra of the immune system, right? With this, I would like to say, really, we can expect a more efficient, effective vaccine, but these vaccines are going to work for only shorter time period because of the virus. Okay, it is going to mutate. It is mutating. Okay, so general comment to everyone, that is it is just a pass on small information. Not only the infectious diseases are really infectious, our smile is also infectious. Yeah, so smile, and because it's infectious. And hope you will try that with your neighbors, your family members and friends. And the choice is really yours, but still we can try to smile. Okay, and I would like to acknowledge and thank Madurai Kamaraj University for this opportunity, and my alma maters, Holy Cross College, Nagarkoil, St. Xavier's College, Palinkote, as well as University of California, San Francisco, and my family and friends. Right? Thank you. You can now get your questions clarified. And here you have my email ID. If you have any specific questions, you can go for it. Otherwise, you can, yeah. Yes, ma'am. Yes. Yes, you can.
Thank you, ma'am, for sharing different dimensions of vaccinology with us.
Yes.
Knowing about the evolution of vaccines was fascinating. Facts about chickenpox party was very interesting, ma'am. Your
Okay.
slides were very engaging and comprehensive. We
Okay.
are grateful to you for providing awareness on Mission Indradhanush, ma'am.
Yes.
Ma'am, no wonder you got the Innovative Teacher Award as you explain things with such ease and clarity.
Thank you.
As ideal vaccines, you are one of our ideal teacher, ma'am. Thank you once again for your informative talk, ma'am.
Yeah. Thank you. Thank you.
Participants, I request you to post your questions in the chat box. Ma'am, shall I read out the questions for you?
Yeah. Yeah. I can also check the chat box. And if,
Oh,
mm,
okay, ma'am.
mm, is there any possibility of reactivation of attenuated pathogen, that is virus? Okay. Earlier days, they were thinking that polio is getting reactivated that way. Okay. Because of differences in storage temperatures and all those things. And so, if at all any person comes and comments that they got developed a vaccine or sorry, a disease after taking a vaccine, what they will be doing is the insurance, you can claim the insurance and you can fight for your case. And so, the sample will be taken from the patient as well as from the vaccine what was administered, that batch also it will be studied, and both the strains will be studied. Is there a chance for this virus becoming new, reverted back in this particular individual, like that for this individual? But the chances of very low. Okay, the chances of very low. It is not always like that. Uh, can we use serum therapy instead of plasma therapy for COVID-19? Uh, the serum therapy and plasma therapy, I think you are the in plasma therapy, technically, to speak to a technical audience, we are aiming at the antibody, right? Antibody of the individual against that particular disease. They have actually it has been against SARS-CoV-2. If a person is being infected, the antibody will might have developed, and that also in the initial stages it will be IgM, and then at later stages it will be IgG. This IgG will stay longer time period in the individual, and so that will be taken and it will be given to the other individual. Even though other people may think that it is just a blood transfusion they are doing, it's not like that. It has been passed in the machines and plasmapheresis is being done, and then the antibodies are harvested and it is being given. What is serum therapy is a old name which was used for in animals, when against the antibodies were raised in animals against some infectious diseases like diphtheria, and nowadays those serum therapy is not given. So plasma therapy is okay, I guess, but that too will not also offer lifelong protection. It is also going to be only a short-term protection. There is one BCG vaccine will be effective against COVID-19 virus. Can you explain? Yes, BCG vaccine is a BCG or tuberculosis, Mycobacterium tuberculosis is itself a general immune booster. So if we read about the France complete adjuvant as well as incomplete adjuvant to boost the immune response, the complete adjuvant has actually the heat-killed mycobacterium. Okay, so this is a general immune booster. So the concept is the people who has actually taken the BCG vaccine, their general immune system is boosted upon, and so they will be really having a good immune response that way against any pathogen. They may be thinking like that. So a certain age group of individuals, those who have not taken BCG, the older individuals, they thought, okay, we can try giving it once again. But in India, whether it is needed, the question is debatable, because we are coming in contact with the Mycobacterium tuberculosis every day, and for sure the person might have been boosted by natural pathogen itself. Okay,